My screen should be shared now. Okay, what are we going to do today? We will revise all the possible experiments that we should revise. Okay, but I want to also make it clear that you can get any type of experiment, not necessarily the ones that you will be revising today, but they're very common. Okay, I'm going to start in order. I did send you a paper 6 summary, and you have a summary of all the paper 6 possible questions that you can get over each lesson. Now, concerning the first two, classification and organization of the organism, in classification, they can get you a dichotomous key. So revise any dichotomous key, or they can ask you simple questions concerning the classification of different organisms. So usually they ask about invertebrates and insects, okay? The comparison between insects and other invertebrates. Now, where is the picture? These are the vertebrates. Of course, this is something you already know about. Now, the invertebrates. Now, the specific points concerning invertebrates, the insects, of course, this is old information that they have 3 pairs of legs, arachnids 4 pairs of legs, and crustaceans 5 pairs and above, or 4 pairs and above. Crustaceans, like the lobsters, the shrimps, they live in water, so they have gills, and they have 4 pairs plus, okay, or 5 pairs plus. They have lots of legs. And which one of them has compound eyes? Insects and crustaceans. And which one does not have antenna? No antenna, zero antenna, are the arachnids. And the crustaceans, they have 2 pairs of antenna. Now, this is significant. So, because they usually ask you about what is really different. The myriapods, of course, these are many, many, many pairs of legs. And they usually ask about the eyes and the legs and the antenna. So please revise those. Okay, you have them here. The antenna, crustaceans, they have 2 pairs of legs, and the eyes, it's the insects and the crustaceans that have compound eyes. Who does not have antenna, zero antenna? The arachnids. Okay, so don't forget that. So this is what you can get concerning paper 6 and classification. Now, the second lesson, which is organization of the organism, of course, the magnification equation. I'm not here to revise it because you know it very well, but the units, you will not convert to micrometers except if they ask you. And 90% of the time, you will not need to convert to micrometers. Why? Because paper 6 is a common paper between year 10 and year 9, the core. And year 9, they don't do micrometers. So don't forget that you will measure everything with your ruler. Your ruler is in centimeters, and you will convert that to millimeters, even if they don't tell you. And even if you will measure something and put the results in a table, the heading of the table, length of plant or length of whatever, between brackets, millimeters. Even if it is in centimeters, you will convert it. Okay? How do you convert from centimeter to millimeter?
Times
Give me
10.
an answer. Times 10. Okay? So the rule of thumb, the standard unit for time is seconds. The standard unit for length is millimeters. You are confused. When do I convert to micrometers? Don't, except if they ask you. And they will not ask you except in paper 4, because micrometers is supplement. It's not core. And paper 6 is a common paper. So they will not ask you to convert it to micrometers. And even if they do, you already know that you will multiply by 1,000. So the magnification equation, definitely you will use it. Okay, it's very easy. Now, the important part, the food tests. Of course, do not step into paper 6 without. Yes.
I have a question. If the يعني لو الريزلت طالع بالمينتس أحوله immediately لسكندز ولا لما يقول؟
No, convert it to seconds. You will keep it in minutes if they tell you the opposite. Like, for example, what do you keep in minutes? The pulse rate, because the pulse rate is beats per minute. Good that you reminded me. The pulse rate, the breathing rate, how many breaths per minute. Okay, so note this down. By standard, the length or the time is in seconds, except if they tell you, or except if the units are in minutes, or they tell you to measure breathing rate. Okay, write it down somewhere. Breathing rate or pulse rate, because the pulse rate is never in seconds, how many your heart beats per minute, and the breathing rate, how many breaths per minute. It's going to come also in the summary. Okay, good question. Now, of course, do not step into paper 6 without revising the food tests and the enzymes and the plants, because I received 100 questions over this question. What do I study for paper 6? Which lessons? Of course, they can get over any lesson, but the common ones, food test, enzymes, counts. And this is what we will revise today. Now, let's be quick. Food tests. Of course, I still get questions over vitamin C. I'll jump to vitamin C, vitamin C, vitamin C, vitamin C. Here, the DC-PIP. And you don't need to know what DC-PIP stands for. You don't need to know. You will start with the DC-PIP in the test tube. The DC-PIP is in the test tube. Isn't the results the conversion of color from blue to colorless? Now, this blue comes from where? From the DC-PIP. So the DC-PIP is in the test tube. And I'm going to add drops of the juice or drops of the vitamin C. Okay? And I will count the number of drops needed to convert the DC-PIP color from blue to colorless. There is no time here. The number of drops. I will add a drop of my juice and I will shake. And I will add a drop of my juice and I will shake again. And I will count the number of drops. Of course, the less the number of drops, the more the concentrated the vitamin C. Please don't get confused. I'm receiving so many questions and answers from investigations. I'm going to put my juice in the test tube. No, my dear. And then I will drop vitamin C. No, you will not get results because the juice has a different color. And you are waiting for a color change from blue to colorless. You have to start with blue. This blue came from where? From the DC. I hope it's clear. Okay, I will not say it again. You have your exam tomorrow. There is no time. Now, the other investigations, let's revise them quickly in case you forgot. Okay, now we will assume that all the investigations or all the samples are liquids. Anything that is a solid, you have to crush it first. Sometimes they try to trick you. They tell you or they ask you, how do I test for fats in seeds? You cannot just add ethanol on a seed. The seed is a solid. It will not absorb the ethanol. You have to crush it with a mortar and pestle. You have the picture here. Everything is there. Okay, you will crush it with a mortar and pestle with a little bit of ethanol so that I dissolve the fats. I will filter it and I will use the filtrate, what comes out of the filtration process. I will use the filtration process for the filtrate in my emulsion test. Let's revise fats. I'm going to add ethanol first, not the water. I will add the ethanol first, not the water. Why? Because fats and water don't dissolve. I will add the ethanol to dissolve the fat, to break down the fat into smaller droplets. This is physical digestion. And then I will add the water. The water and the fat will not dissolve, so it will give me a milky or a cloudy color. Of course, they will not ask you, why do we add fats first? This is not O level, this is AS. But you need to know the order only, that I will add ethanol first, and then I will add the water. If it is a seed or it is a solid, I will crush it with ethanol first, and then I'm going to filter it, and to the filtrate, I will add the water. I'm talking here about the fat test, the emulsion test, okay? So these are the two tests that usually confuse you. I always see mistakes in vitamin C test and in the test for fats. The last time, vitamin C, it's the DC-PIP that we start with. The DC-PIP is in the tube, and then I will add drops of vitamin C, take after every drop, and I'm going to look for a color change from blue to colorless. In the test for lipids, I add alcohol first, and then I add the water, because the alcohol will dissolve the fat or break down the fat, and then I will add the water. The water and the fat will not mix, and they will create an emulsion that is milky or cloudy in color. I hope I'm clear. Now, the test for proteins are very easy. Please study the spelling of the burette and the benedict. They're different, okay? Because sometimes the spelling is horrible to the extent that the examiner or I cannot identify this is burette or benedict. The spelling is a mix between both, and they come up with a third language altogether. So this is not funny, and this is going to make you lose marks. The burette, this is how it is spelled. B-I-U-R-E-T, okay? And the benedict is a different story. The benedict is Benedict. Benededi. So just memorize that, please, because don't laugh, it happens. Now, the test for proteins, the burette. You will add this is the food sample here. We start with the food sample here and the test tube. We'll add the burette. The burette is blue. We will wait a while, and it will change from blue to colorless. Okay? So the positive result is the change from blue to colorless. This is protein. Now, the Benedict, you know it too well. You will heat the food sample. This is the solution to be tested. This is the Benedict. You will put it in a test tube. You will heat it in a water bath. Why water bath? Because it's safer. Can I heat it over a Bunsen burner? Yes, of course you can. But for safety precautions, we use a water bath so that we avoid using a direct Bunsen burner. So this is a safety precaution. And the water bath should reach a temperature of minimum 70 to 80 degrees. I will leave it in this water bath for 5 minutes. And the positive color change is from blue also to brick red. There is a range of colors, green, yellow, but finally it will reach orange or brick red. So memorize the colors. So we're done with the food tests. Okay, now movement in and out of cells is a very common lesson in paper 6. Of course, they always get you the visking tubing. I think you saw it. What is the visking tubing in case you forgot? Who will remind me? What is the visking tubing? Hello?
It's an artificial membrane.
Yes, it's an artificial membrane. And this artificial membrane is semi-permeable. Put in mind, it only allows small molecules. Okay, water. Only water can move in and out, and glucose if it is mentioned. Sucrose, can it cross through? No. Starch, no. Okay, so the big molecules cannot cross. Now they will give you so many options. So many options. Like this, for example, this visking tubing contains starch and it contains the amylase enzyme, and they will leave it in different water baths of different temperature. And then they will check the presence, or they will test the presence of reducing sugars, which is the same as glucose, in the external water. Okay, and they will give you results. Now, what do you expect? That the starch will be broken down by the amylase enzyme, and then the glucose will be produced, or the simple sugar will be produced, and it's going to move out of the visking tubing because it is small, it can go out. And then they will do a Benedict test on the external water, and it should give me a darker color. The more the reducing sugar in the external water, the darker the color result. Okay. Now, if you don't know this concept that the visking tubing does not allow big molecules to pass, you will get very confused. So the only thing that you need to know about the visking tubing, that it is an artificial membrane, like a semi-permeable membrane. It allows small molecules to pass. Which molecules? Water and glucose. That's it. Okay. What happened? Did I lose connection? No, I did not lose connection. Good. Now, concerning movement in and out of cells, plants in water and different concentrations of water. Now, this is so important. I know you know it, but we're here to revise things before the exam. Either, hello, do I have you or I lost you?
Yes.
Okay. So the different possibilities, either you have a plant cell in a concentrated solution, or you have a plant cell in a dilute solution, or you have a plant cell in an isotonic solution. Now, this is a good chance to remember or revise our terms. In a concentrated solution, it's hypertonic or hypotonic. If this is concentrated.
Hypertonic.
Correct, hypertonic. How do we remember it? We will assume that tonic means salt and hyper means a lot. So a lot of salt, concentrated solution. And they like the terms water potential. This is paper 4. But you need to understand it for paper 6. Now you have concentration. Solution means you have low water potential and high water potential inside the plant. The water will move out by osmosis. Very good. This is not our concern now because we are revising paper 6. Our concern is the change in mass. You will measure the mass of this potato before the experiment. You will measure the mass of the potato after the experiment, and you calculate the percentage change in mass. Okay, now we said it 100 times. This percentage change in mass, as long as they ask you, calculate the percentage change, so the answer can be negative or positive. So if it's a negative, keep it negative. Keep it negative. Okay, now it will, of course, be negative if the plant is put in a concentrated solution because the size of the plant will shrink. Now the opposite. If I put this plant in a dilute solution, this dilute solution is hyper or hypotonic?
Hypotonic.
Okay. And the same thing, the water potential outside will be too high, the water potential inside will be too low, or compared to outside, so the water will move in by osmosis. It will swell a little bit and it will increase in size. So the percentage change in mass here will be what? Will be positive, a positive answer. Okay, the final mass minus the starting mass over starting mass times 100. Okay, now there was a student who sent me a question, and this question was measuring the change in heart rate after exercise. After exercise, in other words, someone was exercising, so the heart rate increased. And then he stopped exercising, so the heart rate decreased. And then they asked for the percentage change in heart rate. Now, the final value was smaller than the starting value. And the answer that she got was a negative answer. And it's correct because the heart rate decreased. It went back to normal. But in the mark scheme, the answer was not negative. So she sent me, she told me that the heart rate was high while this person was exercising, and then it went back to normal when the heart rate decreased. And my answer is negative. But in the mark scheme, I have the same number, but without the negative. Can someone tell me why it was without the negative in that case?
Percentage decrease doesn't give the negative.
No, the question was calculate the percentage change in heart rate after and before exercise. After exercise, tell me, your heart rate increases or not? When you exercise, your heart rate is increasing during the exercise.
Yes.
Yes, very good. And then your exercise is finished. It will decrease or it will not decrease?
Will decrease.
Of
It will
course, it will
decrease.
decrease. Of course, it will decrease. You are correct. So naturally, the answer will be negative because we started with a high heart rate and then we ended with a smaller heart rate. But why did the answer has no negative? Because they assumed that your heart rate, yes, it decreased, but it went back to normal. It did not decrease below the normal. That is why in the heart rate, the answer will never be negative. This was only one question that she spotted in an old exam. Okay, it was really, really interesting. So, do you understand here what happened? I'll try to re explain that. I jumped to heart rate, but because I thought that this was very tricky and very important. Here, they asked you to calculate the heart rate during the exercise. Of course, you will have a high heart rate. Okay. And then they said the final heart rate after the exercise, of course, it decreased. So you will have a low heart rate. So calculate the percentage change in heart rate. You will start with the smaller number, let's say 10 minus 20, okay, divided by 10. The 10 is the final heart rate, which decreased because you stopped exercising. So the answer will be negative. You will have a negative heart rate. And she got the number correctly. She did the calculation correctly, but she asked me why in the mark scheme it was a positive value and my answer is negative. Is the mark scheme incorrect? I told her no. The mark scheme is not incorrect. And your answer is also incorrect, not incorrect. Both your answers are correct, but it is illogical that your heart rate will ever be negative. Your heart rate increases during exercise, and when it decreases, it goes back to the starting point. This is homeostasis. It doesn't decrease below the normal. So your heart rate will never be negative. Do you understand that? Your heart rate will never be negative. Your heart rate will never be below the normal heart rate, or you will be dying, God forbid. Okay? So this was one question that came out of 1 out of 10 exams, but still it's better to be safe than sorry. So put in mind that the answer is negative. This is normal. Okay? Except in heart rate. The heart rate can never be negative. Even if your answer is negative, it can never be negative because your heart increases in rate during exercising. After the exercise, it goes back to normal. But it doesn't decrease before that because if your heart rate starts dropping below the normal, then you have low blood pressure and you have to go to the ER. And this is a different case scenario. So going back to my plants. Where are my plants? So we
Doctor,
revised. Yes, my dear.
uh, aren't we supposed to, isn't it supposed to be 10 minus 20 over 20?
Yes, you're right. I'm just discussing in general that the heart rate cannot be negative. Do you understand my concept?
Yes,
No, over the starting. The starting was what?
20.
Oh, the starting was 20. Yes, you are right. You're right. I just want to understand my concept. Put in mind, heart rate can never be negative. Your heart rate, even if it increases, it will go back to normal, but it will never drop below the norm. Now, going back to my plants, we discussed dilute solution, concentrated solution. The important one is the isotonic solution. This is my solution. This is my plant. What does isotonic mean?
Outside is equal to
Outside is
inside.
equal to inside.
Very good. Never please write an answer and say osmosis will not happen. No. Why did the weight of the plant did not change? What's the
The rate
answer?
of
With
osmosis was
water
constant.
potential
Water
outside and
moving in
inside.
is equal to
I did
water
not
The
hear
net
all of you
movement was
because you
equal.
all, yes, net. The net water movement is 0. Okay. What does this mean? This means that there was osmosis. Water is moving in and moving out, but the amount of water moving in and the amount of water moving out is the same. So the net water movement is 0. Okay. Now this can be tricky. Now, how is this represented on a graph? Where is the graph? We discussed this. We'll go back to the red blood cells. Where is the graph? It is here. Here we have a nice graph in which I have the different salt concentrations on the x-axis and the percentage change in mass on the y-axis. And there is a meeting point between the graph and the x-axis. If they ask you when or at which concentration was it isotonic, this is the meeting point between the graph and the uh, grid and the whatever and the percentage change in mass and the x-axis. This is the concentration that was isotonic, which is 0.3. They can ask you the question in different formats. When was the net water movement equal to 0? When was the plant put in an isotonic solution? Okay, the answer is the meeting point between the graph and the x-axis. This means that the water moving in equals the water moving out. Okay, so we don't jump. We're done with plants. We will go back to red blood cells. Now, red blood cells is very easy. If they're put in a concentrated solution, what will happen to the red blood cell?
Become flaccid.
No, no. Do we use the word flaccid with red blood cells? It will, in a concentrated solution, it will shrink.
Shrink.
Yes,
Yes,
it will shrink. Okay, flaccid only with plants, my dear. Okay, isotonic solution.
normal.
Yes, no change in mass. And it is in a hypotonic solution that it will burst because animal cells, they don't have a strong inelastic cell wall. So we're done. Active transport. Active transport usually comes in paper 4, but in case they ask you, it of course needs energy in carrier proteins and against a concentration gradient. Because it is an essay question, it usually appears in paper 4. Now we're done with that. This is the same scenario we're talking about, the different in or the percentage change in mass if you put it in different concentrations. Sometimes they ask you, why do they dry it after it comes out of the water? Why do you dry it first before you measure the mass?
To prevent inaccuracy of mass.
Yes. Okay, to prevent any increase in mass and to obtain accurate results in that case. Now, going back to the results, there are different answers. Why do I repeat? Why do I collect large samples of plants or whatever? So why do I repeat?
Remove anomalous results.
And
Yes. Now, yes, you are all correct. In order to obtain accurate results, we used to use this in checkpoint in year 8. You want to add it, fine. But why do we keep on repeating? For 2 reasons. In order to identify the anomalous and remove it, and to calculate average, not to obtain accurate results. This is too childish now. It's correct, but they don't want this answer. And why do you obtain large samples? In order to obtain representative data, which is real data that represents the real thing. Okay? Now, of course, enzymes are so important. They don't get you graphs now. These graphs, they come in paper 4, but they get you experiments. Now the experiments. We will revise all the possible experiments. The catalase experiment. Now, I noticed that you don't understand that this catalase enzyme, we get it from the plant. This catalase enzyme, its source is the plant tissue. Okay, whether a piece of kidney or a piece of leaf or a potato, fine. So this is a very common experiment. I'm sure you saw it a lot now. You're answering questions. We will test the presence or the different effects of temperature and pH on the catalase enzyme. This catalase enzyme will break down hydrogen peroxide
Dr. Manar, we can't hear you.
Oh, see, I was asking. Can you all hear me and see my screen?
We can't see the screen yet, but we can hear you.
Okay, please, like tell me. Okay, now you can see my screen, I assume.
Yeah.
Yes.
I was speaking here about the catalase enzyme. I don't know if you heard me, but the catalase enzyme, this enzyme is found in living tissue, plants, in a piece of liver, okay? And here we will study the effect of temperature on the enzyme. We will study the effect of pH on the enzyme, and the steps are the same. But of course, we will change the method. Now, the catalase enzyme, we understand it comes from living tissue, okay? So here, design an investigation to study the effect of temperature on catalase enzyme, full stop. They don't give you any previous investigation. They don't give you any information, nothing, because they expect you to know this. This is in your syllabus. So what will you do? You will get the test tube, okay? And you will put in this test tube a piece of potato. And you will put a certain volume of hydrogen peroxide. Good. And of course, you will adjust the temperature. How will you adjust the temperature?
Electrostatic controlled water bath.
Very good. So let's start the normal way. My independent variable, and of course, you will write it this way. Okay. And you will not write IV. You will write independent variable. Okay. So the independent variable, the different, start with the words different, the different temperature. And this is not enough. I have to give at least 3 examples. What are they?
20, 25, 30, 35.
Whatever. Okay. Cold, warm, hot. Fine. And I advise you just to use bigger intervals, like because 20 and 25 are still considered cold. So cold, warm, hot, 20, 60, 100. Fine. Now the dependent variable in this case will be the volume of what?
Gas produced.
Very good. The volume, what is the gas that is produced in this equation?
Oxygen.
Good. The volume of the oxygen produced, and you have to mention either here or in the steps how the dependent variable would be measured. And will you keep on collecting the volume forever?
For 10 minutes. Takes for 10 minutes.
Exactly, for a fixed volume of time. So in case you don't forget, or in case you forget, it's just easier to write it in this step. The dependent variable will be the volume of oxygen produced over 10 minutes using a graduated syringe. Okay, and write it again in the steps, it's okay. So the volume of oxygen produced over 10 minutes. Now I will do my steps. Yes, my dear.
Could we count the number of bubbles produced?
Why? Why count the number of bubbles produced when you can use a graduated syringe? Which is more accurate?
The gas syringe.
Okay, then why do you choose the inaccurate method when you have an accurate method?
All right, thanks.
Okay, so the steps, what will you do? First, I will have to adjust the temperature. I cannot mix the enzyme with the substrate immediately. I first have to adjust the temperature. So I will get those. Yes, my dear.
Well, aren't the temperatures 60 and 80 high enough for the enzyme temperature?
No, it depends on the nature of the enzyme. And then we want to test it at this high temperature because we want to see what will happen when the enzyme is denatured. This is one of the
What
tests.
would be the control?
The control, what will the control be? Who will tell me?
A denatured enzyme or a boiled enzyme.
Exactly, a boiled enzyme. So you can choose 20, 60, 80. Okay, the temperature has to be equal intervals 20, 40, 60, 80. And then one of the potatoes will be boiled altogether. Boiled, there's no temperature, it's boiled, or it's not there.
Doctor, I usually write it 20, 30, 40.
It's correct. You are showing the examiner that you are giving different temperatures. You can do it this way or write cold, warm, hot. Just show the examiner that you will use at least 3 temperatures. Okay, they will not focus on this. They just want, even in the mark scheme, it shows that you are showing, you are using 2 or 3 examples. Just show that you're using examples. Okay, 20, 30, 40, fine. Cold, warm, hot, fine. Just don't say different temperatures and stay silent. Tell them at least 2 or 3 temperatures that are cold, warm, hot, 20, 30, 40, whatever. Okay? Now let's move on. Now, we will not mix the enzyme and the substrate immediately. We have to first adjust the temperature. So we will have 3 thermostatically controlled water baths at the temperature that we choose, whatever it is. And then I will have 2 test tubes in every water bath, one containing the enzyme, and what is the enzyme? What is the source of the enzyme?
Done.
Living
What is the
tissue.
source? Very good, excellent. One contains the potato and one contains the hydrogen peroxide. And in each test tube, we have the same potato, the same size of potato, the same living tissue, everything, and the same concentration and volume of hydrogen peroxide. So 2 test tubes, one containing the potato and one containing the hydrogen peroxide. And they will be put in 3 different thermostatically controlled water baths. We will leave it for 10 minutes first so that they take the temperature of the surrounding water or they reach equilibrium. And then we will mix, okay? And we'll put it back in the water bath so that the temperature doesn't change. And this test tube then should be connected to a graduated cylinder. And we will leave it for 10 minutes and we'll go back and look at the volume. That's it. And you don't have to give results at all. Don't say that the more the volume, the more the optimum temperature. You are right, but they only want the steps. Yes.
Do I have to like do I have to have a potato? Can't I just write like catalase enzyme?
You can just write catalase enzyme, but they expect you to know that it is coming from a living tissue because it's also in your syllabus. Okay, but you can,
Okay,
of course,
can I
it's not
also
wrong.
add diagrams in like my investigations?
Yes, you can. If it's a small diagram within the space provided, you can. It's not going to deduct marks on the actually, it can also even give you marks because it will show the examiner the methodology, the steps. And these are the steps. Very good. Constant factors. We said it as we were explaining. Same size of tissue, same volume and concentration of hydrogen peroxide, same pH, same timing, same apparatus, same everything. And the last 2 sentences that will give you a mark. Safety precautions, make sure that you are careful of the temperature, thermostatically controlled. It's a water bath. It in itself is a precaution. And I'm going to repeat at every temperature and calculate average. Clear? Any questions? And this is what you do when you come to change the temperature. Thermostatically controlled water bath and all factors constant in any investigation of any sort. Okay, I hope you are familiar with that. Now, let's say we will use the same experiment, but I'll change the pH. How do we change the pH of any investigation?
Using
Each buffer.
I will use buff. Okay, so I will do the same steps. Fine. And here I'm going to get 2 test tubes, one with my potato, one with my hydrogen peroxide. I want to adjust the temperature. Okay, fine. I'll put it in a thermostatically controlled water bath, but that here, in this case, the temperature is constant. And I'm going to then mix and I'm going to measure the amount. Of course, I will add something to this mixture. What do I add? The potato and the hydrogen
Catalase
peroxide
enzyme?
and the potato is the catalase enzyme. I will add the pH buffer solution, equal volumes of pH buffer solution, but the only difference is that they will be of different pH. Don't forget that, okay? I can also give examples. I can just say different pH, and this will be enough, but at least 3 different pH, weak acid, weak base, strong base, or I can give values, whatever. Everything is correct. Now, what will be the control in this scenario?
The pH setting.
Neutral. pH is
The
neutral.
control neutral pH without buffer, or I will boil the potato also. Okay, both of them can be accepted, but using a neutral pH is better. So I'm going to add to this mixture the pH buffer of different pH. I will leave it for equal amounts of time, and then I will measure the volume using a graduated syringe. That's it. And the safety precaution: don't touch the pH buffer. Use gloves. Repeat at every pH and calculate average. I think it's clear. Any questions? Where am I? Any questions?
Miss, I have a question.
Go ahead.
So I was doing an exam and it said to determine the effective temperature on the rate of photosynthesis in aquatic plants. So
Okay,
I
we
knew
still
how
did
to do
not reach
everything.
plants. Yes, yes, tell me.
قصدي بس الفوتوسينثيسيس بنقيسها يعني نقول بالظبط احنا مثلا نجيب تست تيوب ونحط بلانج او اسمها ايه نحط سيرنج فيها ولا نقول بس add a syringe او
You will get the plant, you will put it in water because how do I measure photosynthesis? Also by the volume of gas produced. But here, which gas will be produced? Also, it's oxygen gas.
oxygen.
Okay. I'm going to put my plant in water. Good. In this setup. If you don't know how to describe the setup, you can draw it. And this conical flask will also be connected to a graduated syringe. Okay. The same. And show that it is graduated. And then you will change the conditions. You will put it in a different temperature. You will add different pH buffer. You will change the concentration of CO2. This is the setup that we use always when we come to measure photosynthesis. We measure the volume of oxygen produced over a fixed amount of time while changing the environmental conditions. The environmental conditions can be, I will change the concentration of CO2. I will change my pH or I will change the temperature. It's always one of those three. Okay, so the same experiment, but I will change one of those three. I will leave the plant in this condition or light intensity also, the light intensity. Okay, I'm going to change the independent variable. I'm going to leave the plant in this changed environmental condition for 10 minutes to adapt. Fine. And then at the end of the 10 minutes, I will measure the volume of oxygen produced also by a graduated syringe. Is this better?
Yes, thank you.
Okay, very good. And also we'll go back to plants. I still did not reach plants. We're still in enzymes. So we revised change in temperature, changed in pH, and the catalase. Good. Now I want to revise the very, very popular starch and amylase. Okay, this is a very popular investigation, and I think you can see for yourself how common it is. We want to test the effect of temperature on the breakdown of starch using amylase. Sometimes they will not tell you the use of amylase. So what do? The pictures are very clear. Like I'll get two test tubes, one containing the amylase, one containing the starch. You can see two separate test tubes. They're separate because I don't want the reaction to start yet. First, I will adjust my temperature, and I will adjust this temperature by using a thermostatically controlled water bath. I will use at least 3 temperatures: cold, warm, hot. Okay, I'll put it in this thermostatically controlled water bath for 5 to 10 minutes until it reaches equilibrium. I can monitor the temperature using a thermometer. After that, I'm going to mix the two test tubes together and I'll put it back in the water bath so that the temperature doesn't change. Okay, and then I will wait here for a certain amount of time, 20 seconds, 30 seconds, whatever. And then I will take one drop out of this mixture using a dropper. I'll put it on iodine in a spotting tile. These are small depressions or wells that contain iodine. The original color of iodine is yellow-black. And I will put a drop every 20 minutes. Now I will look for the color change. If the color changes from yellow-black from yellow-brown to blue-black, this means starch is present or it has been digested.
Present.
Present.
It is present. So starch is not digested yet. I will have to wait for another 20 minutes to take one drop out of the mixture and put it in the second well. And I will keep on doing that until when do I stop?
Color remains yellow.
No more blue
Very
black.
good. No more black. Very good. And then the reaction will end. And I will keep on doing this for every temperature. And the faster it remains yellow-brown, okay, the faster the reaction. Like if after 20 minutes, it will remain yellow-brown. So this means that all the starch has been digested in just 20 seconds, not 20 minutes. Okay, is this experiment clear? Because it's really, really common. Sometimes they ask you, why don't I add the iodine directly to the mixture?
Because I won't be able to see the color change.
Okay, and because you will ruin the mixture, you cannot then use it for another 20 minutes and so on. I want to get one drop out of the mixture and then get another drop after 20 seconds and keep it without any interference so that I keep on repeating the mixture, the steps until the color remains yellow-brown. Okay, so I never add iodine directly to the mixture. Yes.
Why can't we just put the iodine and wait for it to change color?
يعني قصدي إن
Okay.
it won't ruin the mixture, I think.
It will ruin the mixture
What?
because you will add the iodine and then you will never be able to use this mixture again. You will have to repeat the whole experiment from the beginning. And you will not see a color change. You will not see a color change.
alright, yes, thank
Good.
you.
Fine. So now we're done with that. And you also have here a corresponding graph. Usually this graph comes in paper 4, but still, I'm not telling you to ignore it. Just revise it, scan it. Now the same thing, the same steps here, but we will not use different temperatures. We'll use different pH. Let me erase everything I have here. So plan an investigation to study the effect of the breakdown of starch with different pH. I will not change anything. I will only change the independent variable. What will be the independent variable now?
Different pH.
Exactly. So I will not do any changes. And you have all the pictures here. I don't need a thermostatically controlled water bath because the temperature will be constant anyway. I will not change it. So I will also get two test tubes, two separate test tubes. Why? Because I don't want the enzyme breaking down the starch too soon. I first want to adjust my independent variable. I'll get one test tube containing the enzyme and the buffer. Okay, the buffer, I will use weak acid, weak base, strong acid, strong base. You don't need to give any more examples, or different buffer solutions. Again, weak acid, weak base, strong base. Good. I'll put this in one mixture and I will put the starch in another mixture. And another student told me, can I put the starch with the pH in one test tube and the amylase in another test tube? Of course you can. The idea is that you don't mix them before you adjust the independent variable. So you have two different test tubes. One contains the enzyme and the buffer, and one contains the starch. They're not mixed. After you add the pH and the pH is adjusted, then you mix. You will mix, okay? After you mix, you will take also, so now you have one test tube here with the mixture. This mixture contains the enzyme, which is the amylase, and the starch and the buffer, all three together. I will take one drop out every 20 seconds. Here is the stopwatch. You wait for 20 seconds and you take out one drop and you put it on a spotting tie. And you wait for the color to remain yellow-brown. Once it remains yellow-brown and does not change to blue-black, then the experiment ended. Okay? So it's the same thing, but I will change the independent variable. Any questions over these two experiments? They're really common. I hope it's clear. Now, here you have a note that is not common, but we need to know that any reaction is affected by different factors, by the pH, by the temperature, and also by substrate concentration. Of course, the more the substrate, usually the faster the rate of a reaction until the substrate becomes too much and I don't have enough enzymes to work on the substrate. And here at that point, the rate of the reaction will become constant because the active site here will be a limiting factor. This is not common, but do you understand it? I am increasing the concentration of the substrate. As the concentration of the substrate is increasing, the rate of the reaction is increasing. So in this part of the graph, the substrate concentration is the limiting factor. But at a certain point, it will become constant. I am increasing the substrate concentration and still the rate of the reaction is constant. So this means that another factor is the limiting factor. Usually it's the number of enzymes. I don't have any more enzymes to work on the increasing substrate. Is this understood? This graph? Any questions? Did I lose you or you're with me? No, you're with me. Okay. Now, any questions concerning enzymes before I move on to plants? Hello? Can someone give me an answer?
No, Miss.
Okay. Is everything clear? Good. Now here it's a page reminding you of the mineral deficiencies. This can also be an experiment. I'm going to remove one of the mineral ions and look for the effect of that on the plant. Of course, if I remove the nitrates, it's not going to grow. If I remove the magnesium, it will be yellow, chlorosis. Okay, this is usually common also more in paper 4. Now, the common experiments. Now, the destarching. Before we start, let me discuss that because I also received questions, even though you have it all in the notes, but still. Now, when do I destarch? This is a nice question. And how do I destarch? When do I destarch? When I will test for the presence of starch at the end of the investigation. I'm going to do a certain experiment, regardless of what it is. And at the end of the experiment, I will test for starch. So this means that I have to de-starch first before I do my experiment. Because if I don't, the starch will always be there. And every time I will test for starch, it will give me a positive result. So the question of when do I de-starch? I will de-starch when at the end of the investigation, I will test for starch. So I have to remove it before the investigation. So it does not always give me a positive result. Is this clear? Hello? Clear?
يعني miss, I test for these search when I test for something if efficient in it, it will no more starch will be produced. Or the
When do
effect
I
of
de-starch?
starch?
My dear, when do I de-starch? When at the end of the investigation, when your dependent variable is the presence or the absence of starch. Is this clear?
Yes.
Like if my dependent variable will be the volume of oxygen produced, I don't need to de-starch. If my dependent variable is how much the plant will grow in a certain period of time, I don't need to de-starch. When do I de-starch? De-starch means remove the starch. When do I de-starch? When my dependent variable will be testing for the presence or the absence of starch. If the starch was already there from the beginning, then every time I do the experiment, my results will be positive. So I have to de-starch before. Okay, I hope this is clear. Now, the second question, how do I de-starch? Who will remind me?
Boil.
By boiling the
and
leaf.
ethanol.
No, don't tell me that,
Leave the
please.
plant with no sunlight.
Yes, of course, you will boil the plant, kill the plant. I will not boil anything here. We only boil with enzymes. Your exam is tomorrow, okay? Tomorrow. So how do I de-starch? I will take my nice plant and I will put it in a dark place for 2 to 3 days only. Not forever, not for a week, not for 20 days. I will put it in a dark place for 2 to 3 days. So during this period of time, it will not photosynthesize and it will consume or it will break down the starch that is already present. It will de-starch. It will take the starch that it already has and use it in respiration. There will be no photosynthesis in those 2 or 3 days, only respiration. So because of that, it will break down all the stored starch. So how do I de-starch? I put it in a dark place for 2 to 3 days. When do I de-starch? That's if only I will measure the presence or absence of starch at the end of the investigation. I hope I'm clear. Okay? So this
Didn't
is
we
the
boil
de-starch.
it to break down the
No, we don't
the
boil it. You are confused. We boil when we test for iodine. When we take the plant and test for iodine. Don't get confused. This is testing for starch. You have 3 things that I'm talking about. When do I de-starch? How do I de-starch? And how do I test for starch using iodine in a plant? How do I test? You are confused. When do I de-starch? I will de-starch if I will test for the presence of starch at the end of the investigation. How do I de-starch? De-starch. How do I remove the starch from the plant? I put it in a cupboard for 2 to 3 days. You are confused between this and how do I test for starch at the end of the investigation? I did my investigation and I will take a leaf out and I want to see this leaf contains starch or not. I will do some boiling here first in order or before I add the iodine. Are you with me? Did you understand where the
Yes.
confusion is? Okay, thank you so much.
Yes.
Okay, good. So this is what was confusing you. Here we will do those steps that we will discuss in a minute after the experiment is finished and I want to test for the presence of starch. So what do? I have a problem. This leaf is covered by a waxy layer. I cannot simply add iodine on the leaf because when I do, the iodine will not penetrate and I will not get results. It's as if putting iodine on air, on nothing. I want to put the iodine on the leaf and I want it to penetrate the leaf and I want to see a color change because I want to know if the leaf contains starch or not. So I cannot just simply add iodine on the leaf. I have to do some things first. What do I do in the first step and why? Who will remind me?
Doctor, how are we testing for starch if we just removed all of it في ال starch?
Habibti, didn't we put the plant through a
Add
particular
ethanol?
investigation? 1 minute, I
Add
have to
ethanol.
answer a question. didn't we put the plant through a particular investigation?
ده after destarching يعني.
Yes, حبيبتي. Look, my dear. Look, my dearest. First, I have a plant. I want to test for the presence of light, for the effect of light intensity on photosynthesis. So step number 1, I'm going to destarch the plant. I will put the plant in a dark place for 2 or 3 days so that the plant has no starch. Step number 2, I will go through my investigation. We said that we will investigate the effect of light intensity on photosynthesis. So I will do my experiment. I will put my plant in a water surrounding, and I will connect it to a graduated syringe, and I'm going to change the light intensity, and I will do this by changing the distance. So I'm going to give a chance for the plant to do photosynthesis. Okay. Very good, my dear. And then the third step, okay, I want to know, while it was in the experiment, did it produce starch or not? This is when I will test for starch. Okay, my dear?
Oh, yes, thank you.
Okay, so I hope this is clear for everyone. We do it in steps.
So
First,
that's
I'm going to
when we are testing or doing the experiment of a limiting factor بتاع الplants بعمل destarching الأول.
exactly. That's if at the end of the investigation, I will be testing for starch. So 1, I will remove the starch. 2, I will put the plant through a certain experiment. And this experiment will involve photosynthesis in one way or another, but I'm going to cause a limiting factor, just like you said. And then at the end of the investigation, I will test for starch to see when I added a limiting factor or when I played around with the limiting factor, did the plant photosynthesize or not? And how do I test for photosynthesis? Did it produce starch or not? There are 2 ways to test for photosynthesis. If it produced starch or if it produced oxygen. If it produced starch, I have to do this. If it produced oxygen, I will use a graduated syringe. Better? Hello? Is it clear?
Yes.
We only destarch if I decide that at the end of the investigation, I will test for the presence of starch. If I decide at the end of the investigation, I will measure the volume of oxygen produced, do I need to destarch?
No.
No. I hope it's clear. I don't want you to memorize anything. I want you to understand what I'm saying. I'll say it for the last time in case someone is confused and they're too embarrassed to say. I want to do a certain experiment to test photosynthesis. Okay? I have certain limiting factors: light intensity, carbon dioxide concentration, what else? pH, temperature. I will change one of the limiting factors, or I'll change one of the environmental conditions, and I will see if the plant photosynthesized or not. Okay, how do I test for photosynthesis? There are 2 ways. Only 2 ways. Either I will measure the volume of oxygen produced at the end of the investigation, or I will measure the absence or the presence of starch. If I will measure the volume of oxygen produced at the end of the investigation, I will use a graduated syringe. And here I don't need to destarch because I'm not looking for starch anyway. But if I'm going to check the presence of starch at the end of the investigation, then I have to start with a plant that has no starch. I have to destarch in this case. So it's your decision, or I mean, it's the decision of the question. Is this clear? Hello? Is this point clear? Good. Now, measuring the volume of oxygen is very simple. I'll use a graduated syringe. How do I destarch? I will put my plant first in a dark cupboard for 2 to 3 days. I will make sure that all the starch is gone. And then I'm going to carry out with my investigation. And at the end of my investigation, I will test for the presence or absence of starch. I will do the following. I cannot simply add iodine to the plant because the iodine will not penetrate the plant because of the waxy cuticle. So I want to remove the waxy cuticle. How do I remove this waxy layer? What do I do?
Boil the leaf in water.
Yes, I will boil it in water, just like you see in the picture, for 5 minutes. So I remove the waxy layer, I kill the enzymes, I will remove anything that will prevent the penetration of the iodine. Now I have another problem. The leaf is very dark green. I cannot see the color change. I want to remove most of the chlorophyll. So what do I do in the second step?
Add ethanol.
Very good. I'm going to boil it this time, not in water, but in ethanol. And because ethanol is flammable, do I provide an open flame?
No.
Do I expose the ethanol? No, this is a health hazard. The ethanol can burst in your face. So I'm going to put the plant in ethanol and I will put the test tube in boiling water or hot water, so that the ethanol boils. The ethanol will boil very quickly because it's very sensitive. And the chlorophyll is going to move out of the leaf and the leaf will be very, very light in color. Okay, now I have a leaf. I will start with a leaf, or I will work with a leaf that has no oily layer, no waxy cuticle on the surface, and it is very light. Only then I can add the iodine. First, I will dip it in water because it will become very, very brittle in the ethanol. The ethanol makes it very dry. I'll put it in water to make the leaf soft. So this step is to make the leaf soft. And then the last step, I will put my leaf on a white tile. Why do I use a white tile?
To observe color change easily.
Very good. And then finally, I will add my iodine and I will notice the color change. If the color changes from blue-black, then this leaf did photosynthesize during my experiment. If the iodine did not change to blue-black, then my leaf was unsuccessful in photosynthesizing during my experiment. I hope it's clear. Anyone is confused? Is anyone confused?
Can
Tell
you
me.
repeat
Don't be
the last step?
the last step. This step, when I will put it in water to soften the leaf.
Yes.
Okay. Now, when I boil any living tissue in ethanol, it will dry it because ethanol absorbs the water. It will make the leaf like flakes. They will break easily. I cannot even put a tweezer on the leaf because the leaf will break. It's like the leaf is going to be very brittle. You know, like the dry leaves that you see on the floor. If you hold the leaf will break. So we want to avoid that. So we will just dip the leaf gently in water, in warm water, so that it becomes soft and I can handle it. That's it.
Okay.
Clear?
Then we will test it for we'll add the iodine, right?
Yes. And then we add the iodine and look for the color change. Good.
Okay.
Now they do ask you, don't ignore it. Yes, it's not common, or actually it is in the recent years, so don't ignore it. Now, the difference between safety hazard and safety precaution. Safety precaution means, how do I protect yourself? How do you protect yourself? I hold the hot instruments using using a tweezer. I'm going to use a water bath so that I don't use an open flame with ethanol. Safety hazard, what is dangerous? The ethanol is dangerous because the ethanol is highly flammable. It can burst next to a flame. Okay, we already discussed that. Is everything clear?
Miss
Hmm.
هو how will the plant photosynthesize مش عارفة ايه during my experiment و I remove the chlorophyll.
Who said that I removed the chlorophyll? Did I remove the chlorophyll? I just removed the
مش
starch.
احنا
The
حطيناها
chlorophyll
في
is there.
ethanol؟
You put it where?
مش احنا حطيناها في ethanol؟
Okay, you mean after the experiment, not during. Your question was, how will my plant photosynthesize during the experiment? During the experiment, the chlorophyll was still there. I removed the
ما هو
chlorophyll
قايل
after the
ما
experiment
هو قايل
so
وهي
that
لسه
I test for the presence. After the experiment, it will not photosynthesize.
I
After
know
the
هو
experiment, it should have photosynthesized, and I am looking for the starch. I just want to see it clearly. I want to
okay.
see the color change clearly. So I'm going to remove the chlorophyll so that the plant is not very, very dark green, and I will see a color change when I add the iodine.
يعني it will photosynthesize before the experiment.
During the experiment, my dear, during the
يا
experiment.
مس في
It
step
will
2 يا مس مكتوب we remove الchlorophyll مش الchlorophyll ده essential
This is another experiment, حبيبتي. Another experiment. We remove the, we remove the chlorophyll after the experiment. This is not an experiment. This is a test.
أيوه
This is a test. This is a test. You're confused. This is a test. I want to test for iodine. I want to test if the starch is present. I have to remove two things before I add the iodine, the waxy layer and the chlorophyll, before I add the iodine.
okay thank you.
Clear? So this is not an experiment. The experiment is finished. This is the testing for the starch after the experiment. Are you with me, Selma? Is it clear?
Yes.
Better now? So we're not photosynthesizing here. The photosynthesis is finished and the experiment is finished. Okay, was the experiment successful or not? Did the plant photosynthesize or not? I want to add iodine. I'm sorry, I cannot add iodine directly. I have to remove the waxy layer and I have to remove the chlorophyll. So I can see a color change. Okay?
Okay, thank you.
You're welcome. Now, a second experiment altogether. Is chlorophyll important or not important? Again, I'm going to choose this. I will not de-starch here. I'm going to get a plant with no chlorophyll. Now, how do I do that? I don't do anything. I just choose a leaf that has white parts and green parts. And this type of leaf is called the variated leaf. Okay? And then I'm going to no, here, if I will test for starch, I will have to de-starch. Put in mind, anytime I have to test for starch at the end of the investigation, I will de-starch. So step number 1, I will choose this leaf that has different colors. If it is green, then it has chlorophyll. If the part of the leaf is not green, it's whitish, then there is no chlorophyll. Okay, I removed chlorophyll from parts of the leaf. And then step number 2, I will de-starch. I will put it in a cupboard for 2 to 3 days. So the starch is gone. So now I have some parts of the plant, no starch, no chlorophyll. And then, okay, now I will expose it to normal environmental conditions and see if it will photosynthesize or not. I will put it in the sun. Please do photosynthesis. I will leave it for 2 days. I will come back. I will take this leaf and I will test for the presence of starch. Of course, I have to prepare it before I test for starch. I will boil it to remove the waxy layer, and I will also add it and boil it in ethanol to remove any chlorophyll that is there. And then I will add my iodine. Is this experiment clear? So we did the same thing, but here we used a leaf that is parts green and parts without color because here we're testing if the chlorophyll is the limiting factor or not. Are you with me?
Yes.
Okay. Now, if we want to test for is light important or not? So number 1, and at the end of the investigation, I will test for starch. So if at the end of the investigation, I will test for the presence or absence of starch, what do I do before the investigation?
De-starch.
Thank you so much. I will de-starch. Okay. And then step number 2, I will cover it with aluminum foil, just like this. And why do I choose aluminum foil?
To reflect light.
Very good, to reflect the light from different parts or from certain parts of the plant. And then after I cover it with aluminum foil, I'll put it in the sun. Why do I put it in the sun?
Be able to photosynthesize.
Exactly, so that it will do photosynthesis. I will leave it for 2 days and then step number 4, I will come back. I want to see which parts exactly were able to do photosynthesis and which parts were not able to do photosynthesis. The one that was exposed to light or the one that was not exposed to light. So how do I know? I will prepare it. I will boil it in water and then boil it in chlorophyll and in ethanol to remove the chlorophyll and then put it in water again to make it soft. And then add iodine on this part and add iodine on this part. Which part do you expect will give me a positive result? The part that was exposed to light or the part that was not exposed to light?
The part that was
Exposed.
Okay, very good. So nice and easy. These are easy experiments. Here, I want to test for another limiting factor, the presence or the absence of CO2. So first, I'm going to get the plant and I'm going to de-starch. And then step number 2, I'm going to expose it to this environmental condition. I will put a cover and I will put here soda lime, and they will tell you, you don't need to memorize those things because the soda lime will absorb any CO2. It will remove any CO2. In this situation with no CO2, I will put it in the sun. So I want to see, will it photosynthesize without CO2 or not? After I put it in the sun, I will come after 2 days and then I will test for the presence of starch. But before I do that, I have to prepare it. I will take 1 leaf. I will first boil it in water to remove the waxy layer, and then boil it in alcohol or ethanol to remove the chlorophyll so I can see, and then dip it in warm water so it's nice and soft. And after I finish all those steps, only then can I test for the presence or absence of starch by adding iodine. Is what I'm saying clear?
Yes.
Any questions? Good. Okay, so I hope now you understand. If I will test for the presence of starch at the end of the investigation, I de-starch. If I will test for photosynthesis by measuring the volume of oxygen produced, I don't need to de-starch. Okay, and this is also another investigation. Again, we remove carbon dioxide, we add carbon dioxide. I play around with the independent variables, and in the end, steps are the same. Now here is the second method for testing for photosynthesis. Is oxygen produced? Here, do I need to de-starch?
No.
Exactly. Now you understand. I don't need to de-starch because I'm not going to be testing for starch anyway at the end of the investigation. I'm going to be counting number of bubbles or using a gas syringe if possible. Or I can test if this gas is oxygen or not using the glowing splint. This is the test for oxygen. It's not very common, but you need to know it. I'm going to get out like this oxygen is being collected in this test tube. I want to know if this gas is oxygen or another gas. What do? I'm going to get out the test tube and very quickly I'm going to insert a glowing splint. If there is oxygen here, what will happen to this oxygen?
The globe splint will relay.
Yes, very good. Okay. Now we understand limiting factors. Now here I am studying light intensity. How do I change the light intensity?
Using a lamp
Distance of lamp.
Very good. And why is this water tank in between the lamp and the plant?
to avoid
To
changing
absorb
the heat.
the
To absorb heat. Okay, very common and very easy. And this is a better picture showing you the graduated syringe. Okay, now here we're changing the temperature of the water. We use different water baths or we use different heaters and we adjust the temperature of the water. And we will monitor the temperature of the water using a thermometer. And again, we will count the number of bubbles that is inaccurate, or we will use a graduated syringe. And this is a better improvement. I'm going to use here a beaker of water to absorb the heat. So they play around with the independent variable. Now I'm done with the plants and all the experiments related to plants. Is there any question? Can I move on?
Yes.
Okay, good. Now, nutrition, usually they get experiments on enzymes, honestly. Rarely do they ask about the nutrition needed, but in general, children need more food than adults because children are growing, and pregnant females, they need more food because they have a baby in their stomach that needs calcium and protein and fats, etc., etc. And a breastfeeding female actually needs the most energy because she is producing milk, and this milk is rich in proteins and fats, etc., etc. Okay, so this is too easy and too common. Now, also more plants, the location of the vascular bundle. Now, this is important. We have three organs in a leaf. We have the root, we have the stem, we have the leaves, and we want to know the vascular bundle or the xylem and the phloem in the three organs. In the stem, the vascular bundle is always located in a ring like this. The xylem is always directed towards the inside, and the phloem is always towards the outside. So if I will draw a simpler picture, this is my vascular bundle arranged in a ring. Okay, and the xylem is always towards the inside. This is the xylem, and the phloem is towards the outside. They can get you a picture like this and tell you, is this the stem or the root? How do you know the arrangement of the vascular bundle? If the vascular bundle is arranged in a ring, it's a stem. Now, if it is arranged in the center, this is a stem or a root?
A root.
Root.
It's a root, okay? And where exactly is the xylem and the phloem in this root? The xylem is always in the center of the center. So this is the root. The vascular bundle is in the center. The xylem always looks like a flower, and it is the central tissue. This is the xylem. And why is it a tissue? Because it is made up of similar cells that carry out a similar function. It's an organ because it's made up of different tissues that have a particular function. Now, the last organ in the leaf, in the plant, is the leaf. This is the cross section of the leaf, and this is the vascular bundle. Where is the xylem? The xylem is always towards the upper surface of the leaf, and the phloem is always towards the lower surface of the leaf. So memorize their locations. Okay? Now, this is a simple investigation where they will put a piece of celery. You know what is celery? Karafs? Okay, they will put it in different solutions of different color. And because there is transpiration and absorption of water, the water will be absorbed up the xylem. And then they will look at the cross section of this ciliary and they will find that the color of the water is arranged in a ring. So this proved that the vascular bundle in the stem is arranged in a ring. This is a very simple experiment. Any questions? Now, we did talk about wilting and flaccid. It's just a different term. Please, you will draw, of course. Draw a leaf. Practice drawing it. Maybe they will ask you to draw it without a previous picture. We talked about drawings. Now, chromatography. It's not very common, but it did come in one of the recent exams, and I'm happy that you took it in chemistry. Now, what is the idea about chromatography? They will extract some pigment from a plant from any source, and they will put it on filter paper. Now, for starters, this filter paper will have a line. This line is made up of pencil. Why pencil? Or I draw it with pencil.
Not soluble.
Excuse me?
It is not soluble in water.
Yes, correct. It's not correct. It's not soluble in water, so it will not be dissolved and ruin my experiment. And then I'll put this filter paper in water or alcohol, and the level of the water or the alcohol should be below this line. I'm going to put on this line my pigment. Let's say I got it from a leaf. And then what will happen is that the alcohol will be absorbed by the filter paper, and then the alcohol will move up the filter paper. And as this happens, the pigments in this drop will separate. Now, the one that is very, very close to the line is the heavier pigment or the lighter pigment?
Heavier.
The heavier. And the lighter one that travels quickly will, the one that will travel the most. Okay. Now, this is a common one. How do you investigate the effect of stomata on transpiration? Now, transpiration is a different story. Transpiration is the loss of water vapor from plants. How do we investigate transpiration? It's always by the weight or the mass of the plant. I will measure the plant before and I will measure the plant after. And the more the weight drops, the more the water loss. I hope I'm clear. So I'm going to investigate here the distribution of stomata. I will get leaves from the same plant, from the same source, the same size, and I'm going to hang them like this, okay? And I'm going to use Vaseline to cover my stomata. One time I will cover the stomata below the leaf. One time I will cover the stomata above the leaf. One time I will cover above and below. And one time I will not cover at all. This will be my control. This will be my control. Now, we will measure the mass of the leaf before the experiment, and then I will add my Vaseline, and then I will hang it like this for a few hours, and then I will measure the final mass. Which plant do you think will lose the most water or will have the greatest percentage decrease in mass?
No petroleum jelly.
Yes, it's not covered, so it will lose water. And which one will have the least percentage decrease in mass?
Lower surface area cover.
Yes. Okay. Now this shows me that you understand the experiment because most of the stomata in most plants is at the bottom. So when I cover the bottom of the leaf with Vaseline, least transpiration will happen and least decrease in mass. Okay. The potometer. Potometer also measures transpiration. Transpiration is the water lost. We cannot measure how much water vapor escaped into the air. This is hard. So how do we do that? We measure the change in mass of the plant and we change the environmental conditions. Now, changing the environmental conditions, you need to know how it's done. If I will measure the mass of the plant before, and then I'm going to change the humidity. How do I change the humidity around the plant?
The use of humidifier or dehumidifier.
Yes, a humidifier is correct. Or easier, I'm going to cover it with a plastic bag. When I cover it with a plastic bag, the humidity would increase or decrease?
Increase.
Yes, good. And as the humidity increases, how does this affect transpiration?
Decrease.
Correct. Good. Very good. Now you're familiar with that. How do I change the light? Now this is so easy.
Lamp.
Correct. The distance from a lamp. How do I change the wind speed?
Fan.
Fan. Very good. Perfect. How do I change the temperature? I don't put it in a water bath.
Digital heater.
A heater. Okay. The temperature of the surrounding room. Very good. Now this is a bubble photometer. How do I calculate the rate of transpiration?
How much air bubble moved?
Very good. Over the distance the bubble moved over time. Very good. Excellent. Okay. Now, why when I change the independent variable, do I give or do I wait for 5 minutes before I start?
For the experiment to adapt to the independent variable.
Very good. For the plant to adapt. Good, good, good. Lovely. Okay, what else? What else? What else? We said all that. We also said that. Now, those 66 pages, I'm not telling you to memorize them. No, I'm telling you to understand them. What else? What else? What else? We did talk about the humidity. Yes, measuring pulse rate. Now, this is only in minutes. We said that. Pulse rate is the number of heartbeats per minute. Good. How do I measure the pulse rate? You need to provide a method of pulse rate investigation if you have an investigation question. The picture is very obvious. I'm going to put my fingers, those 2 fingers, on an artery, not on a vein. Why do I put it on any artery in the body? What am I trying to sense or feel?
Pulse.
Heartbeat.
Pulse. And does the vein have pressure? Does the vein have a pulse?
No.
No. Okay. So I'm going to put my fingers on any artery, any obvious artery. You don't have to know the location and you don't have to know the names. I'll just mention that I'll put my 2 fingers on an artery and feel the pulse, which is the number of times the artery will pulse in minutes. Minutes. Okay. The heart rate is beats per minute. And as we said, the heart rate can never be negative because even if you exercise and the heart rate increases and then it will go back or decrease, yes, it will decrease. We did not deny that, but it will go back to normal. Your heart rate never drops below the normal, or then we have a situation here. Now, the stethoscope can also be used to measure the pulse rate, but you hear the valves closing and the electrocardiogram, of course, but we usually don't have it accessible when we are doing investigations in the playground. We don't have a nearby electrocardiogram. Now, this is important here when you have investigations concerning health human subjects. I will use either the same human, which is the best, but before I change the intensity of the experiment or the intensity of the exercise, I have to make him rest. The heart rate has to go back to normal. Or I will use a group of human subjects, the same age, the same gender, the same health status, and this can never be 100% accurate. Okay, so using the same human is best. If you receive a cardiogram picture like this, every curve or every crest, this is 1 heartbeat, and here the heart is relaxing, here is 1 heartbeat. If you are asked to measure the heart rate from this electrocardiogram, you will count the number of beats. This is 1, 2, 3, 4, 5, 6, 7 beats per minute, for example, if this is in a minute. Okay, if it's not, you have to get it per minute. So when do you use minutes? Heartbeat and breathing rate. Okay. Now, you will not be asked about the percentages of in and out air. This is paper 4, but this can be paper 6. How can you measure respiration? Using a respirometer, this is more accurate. Again, breathing rate per minute or the up and down of your chest every minute, which is not very accurate, counting the number of times the chest moves up and down per minute. What else? What else? Hydrogen carbonate. You need to know the colors. Hydrogen carbonate is an indicator that measures the percentage of CO2 in liquids. If it is orange, then we have an average amount of CO2. High CO2, it becomes yellow. Low CO2, it becomes purple. So memorize the numbers. Do I still have you? Or we lost each other? I still have you. Promise I'll finish soon. I know that you have an exam tomorrow. What else? Respiration in yeast. Again, this is so simple and the same concept. If I will change the temperature, I use different thermostatically controlled water bath. If not, I'm not going to choose or use a water bath anyway. Now, let's jump. Why do I put an oil over the yeast and the glucose? This is when I am testing for aerobic or anaerobic. Anaerobic. Very good. Okay. Now, this is coordination. I know you revised it for paper 4 and it can come in paper 6, but in paper 6, they will ask you about the clinostat. why one set of plants were put in the independent variable, whether light or gravity, and one set of plants were rotating slowly?
علشان to cancel gravity.
Yes, to cancel out the effect of gravity or the light. And this is the
Constant.
Control.
the control. And I always have a control to compare. Good. Now, seed germination, they usually ask you, yes, they do ask you about plan and investigation, where you will study the effect of one variable over seed germination. Here, I will have seeds that I either put in a pot and I add water, or I have seeds that I put in a petri dish and this cotton is wet with wet cotton. Okay, so it's your choice. Either I will plant it in soil or I will plant it over wet cotton. And I will change the independent variable as they want. How do I change the temperature? I will not put it in thermostatically controlled water bath. Okay, the seeds will be soaked, they will die. I'm going to put the seeds in a room of different temperatures. So here I will use a heater. If I will change the pH, I will add different pH buffer solutions in the soil or in the petri dish. Now, the common question, how do I test for germination? I will keep the seeds in this situation for a few days, and then I'll go back and I will count the number of seeds that germinated. If you want to measure the length of the part that germinated, it's your choice. But it's more accurate just to count the number of seeds that germinated, the number of seeds that already erupted into a small baby plant. You can see it and you can simply count it, and that's it. Okay, let's show you a picture of a germinating seed or germination. 1 minute. Okay, so you understand how germinating seeds look like. Here. This is a real picture of a germinating seed. Okay, or they look like this. These are seeds that germinated. It's difficult to measure the length of the part that germinated. The part that germinated is the plumule. It's very difficult to measure the length. See? This is very short in millimeters. So you will measure this. It's hard. So it's easier to count the number of seeds that germinated. Is the picture clear now? I hope it is. So we're done with germination. The dry mass of the seed. Here, we know very well the germination starts with respiration. That's why the dry mass of the seed will decrease and then it will start increasing. Why does it decrease first? Because it will respire first, consuming the starch that is already present in the seed, and then it will increase when the seed starts photosynthesizing. It's not common, but I always say it's better safe than sorry. This graph, you will not get it in paper 6. Just have a quick look at it because you will be asked to plot a graph like this in paper 4. Now, we're done with all the experiments and we revised the graphs yesterday. Any questions? Any questions concerning any experiment of those?
No.
Is everything clear? Do you want me to revise anything? Anyone has any question again for the 10th time?
No.
Okay. Best of luck, and I'm here the whole day. If you have any question, just like you do, send me a picture, and when I see it, I'll answer you. Fine? Good luck. Bye-bye.